ribosomal frameshifting occurs when ribosome changes its reading frame. And so as a result, what is happening is that you have two open reading frames which are overlapping. And as a result of ribosomal frameshifting, you could produce a single protein from these two overlapping open reading frames. Normally, ribosomal frameshifting is inefficient. And say it's quite frequently utilized by viruses, including SARS-CoV-2, which is a source of current pandemic. Because it is inefficient, it competes with the standard translation, so that because of this possibility of making a standard protein and a frame-shifted protein, you get two proteins, and you get them at a particular ratio. And so in case of, say, SARS-CoV-2, there is a very, very long protein, polyprotein. They call it polyprotein, the longest translated region in the virus. Somewhere in the middle, there is a site of ribosomal frameshifting there. And so we think that the reason why it is there is because the virus needs proteins upstream of frameshift site at a higher concentration than those downstream of ribosomal frameshifting, because those would be synthesized only as a result of this ribosomal frameshifting. But ribosomal frameshifting also has been found in genes from practically all organisms, including humans. There are very, very few cases in humans. Only 5 human genes are known to use ribosomal frameshifting. But it is possible that there are more. We just haven't discovered them. And ribosomal profiling could be very helpful at identification of such cases, because ribosomal profiling allows not only to find where the ribosomes are and what regions are translated, but also detect the reading frame that is being translated. And so once you find a switch or change in the translated reading frame, using ribosomal profiling, you may investigate it further to see whether these are just two independent translations that in different frames, or whether there is a ribosomal frameshifting occur there.